Why don't we give people another 2 minutes and then we'll go ahead and get started.
Okay.
And Veronica, why don't you just move down to the first slide that shows the workshop organizers, and we'll start there. Excellent. Okay, I think we will go ahead and get started. I want to welcome everyone. Good morning, good afternoon to the virtual workshop on the analysis of DArTseq fingerprinting data. This workshop, I want to thank the sponsors, CGIAR, the SIP, and the Crop Trust. This has been part of a ongoing effort for two separate projects, which I'll describe just in a little bit. But first, I want to make sure that everyone is on board for this short 2-hour workshop. It's going to be fairly technical, but how it's been organized is that you can take all the information and cut and paste it. We will send around the URL to the workshop. If you don't already have it, we'll resend it at the end of the workshop. And let's go ahead and get started with the introduction of the projects that this workshop is part of. I can get the next slide, Veronica. The first project is the Seeds for Resilience project, which is a 5-year project. It's funded by the German government and managed by the Crop Trust. It's providing technical and financial support to enhance conservation and sharing of plant genetic resource crop diversity from 5 countries: Ethiopia, Kenya, Ghana, Nigeria, and Zambia. This workshop focuses on the collection of about 250 sweet potato land races from Zambia. Now, we're not going to report on all 250 land races, but we take a subset, and we're going to use that as the basis for this workshop. All these land races were collected in Zambia and then shipped to SIP Nairobi under the SMTA from the International Treaty for Plant Genetic Resources for Food and Agriculture, and then put into in vitro. We're in the process of also shipping all of these land races to SIP Lima for long-term backup and inclusion in the in-trust sweet potato collection. Once they are in SIP Lima and phytosanitary clean, they will be available under the SMTA from the International Plant Treaty to any researchers for research, breeding, any researchers or breeders for research, training, and breeding. The next slide, please. The second project that this workshop is being organized for is the Darwin Clean and Share project. This project was part of the Darwin Initiative funded by the UK, and it's for the collection and long-term conservation of land races from both Madagascar and Zambia. And in this respect, it's piggybacking onto the material from Zambia. Where the Darwin project separates in Zambia and is 100% for Madagascar is that the material was collected and put into in vitro at SIP Nairobi. This material was then phytosanitary cleaned, 30 of the land races, and the phytosanitary clean material was repatriated to the country of origin, that is Madagascar and Zambia, and it's currently undergoing multiplication and will be distributed as disease-free vines back to farmers. The Darwin project will further then assess the farmers' benefit from planting the disease-free material. And the other aspect of the Darwin Clean and Share project is it is a cornerstone of a global plant cryopreservation initiative that we are currently getting started, which will conserve in plant cryopreservation long-term, that's in liquid nitrogen, all of this material. Next slide, please. As far as DArTseq genotyping is concerned, both projects had their material genotyped by diversity arrays in Australia. What the genotyping provides us is an opportunity, as you'll see in this workshop, to do some diversity analysis, that is, compare one land race to the other. We can identify unique land races, and the flip side of saying that is we identify duplicate land races, and it provides a lasting identity for these land races into perpetuity. This workshop will demonstrate the preliminary method that we use at SIP for the analysis of DArTseq data using the sweet potato or a subset of the sweet potato land races from Zambia as an example. The DArTseq genotyping from Madagascar is currently ongoing, and once we get the complete analysis of the DArTseq data from both countries, we will be able to compare the land races from both countries, both on a diversity basis, and to start asking questions about where those land races are most similar to, using the entire INTRUST sweet potato collection, which is at SIP. So at this point, I will turn the the workshop over to Veronica Valencia, who will introduce us to the analysis of the DArTseq markers. It's all yours, Veronica. Thank you.
Hi, hello everyone. I am Veronica. So I would like to start explaining a little bit about what is the diversity arrays technology. We are currently using a marker called DArTseq. So Diversity Arrays is a company, an Australian company that uses their proprietary DART platforms to provide a service of markers for any organisms, in this case for our sweet potato. They have large bioinformatic capacity and are constantly developing new tools to aid in marker development and analysis. So this company created these markers. They started as a DART. They have something called a DART platform that provides this generic and cost-effective genotyping technology that detects all types of polymorphisms. So it was invented by Andre Killian to overcome some of the limitation of other molecular markers. So we're using this marker because of all of these benefits that they have. A little note here, because there is a bit of a difference between DART and DArTseq. And if you want to read something about this marker, you should go and look for DArTseq and not just DART, because darts were created in 2001, and the DART name remained, but now they are working with DArTseq markers because they are using a technology of reduced representation sequencing. So DART provides three types of files. They give you 3 reports. So when you download the information they give you, they provide 3 different data sets, something called SilicoDART, that is a marker of presence and absence, and 2 files for SNPs, or single nucleotide polymorphisms, one in a 1-row format and the other one in a 2-row format. So in this workshop, we are going to be focusing on the SilicoDART markers. And if you want to know a little bit more about the SNPs, you can do it. I am giving a little bit more information after the workshop, but today we're going to use only SilicoDART. So these SilicoDART markers are dominant markers presence and absence, and they are obtained by the successful restriction of genomic DNA. So a 1 in this dataset represents a presence, a 0 an absence, and missing data is represented by a hyphen. Sometimes in other datasets, you will have a 9 or a 99, but for DArTseq markers, it's normally a hyphen. So a little bit of why it's called silico DART, because silico comes from in silicon. That means carrying out experiment on computers or via computer simulation. That is why it's called silico DART. So when you download your report, the silico DART report, you will see a table like this. If you open it in Excel, you will see this format. So to explain the format, we are giving it some color, and this is the color report, but you will receive it like this, in just everything in white. In these examples, you will see in blue that these have marker IDs and sequences. In yellow, you have information about mapping position and scores. And in green, you will have some marker stats like call rate, reproducibility, etc. So if you want to know the parts of these reports, you will see that the marker ID, because every marker, every silico DART marker has a unique name that is represented by a number in this column called clone ID. And if you want to know the names of the samples you sent, the sample ID is in this part. Right? So if you notice, we have some asterisks here. This is not useful information. It's just for the program to know how many of them they need to skip. Right? So in this case, we have 6. And why is needed this? Because it's the same length of the information of the plates. This information is very useful for us to locate samples, but sometimes for the programs we use, we need to skip all of this. So presence, absence, and missing data. If we zoom and we want to know the data we have every sample, and we have here for the first marker is a 1, for the second is a 0, and you will see these hyphens as missing data. So if you want to see a little bit better about the blue columns, you have here the clone IDs, the allele sequences, and the trim sequence. And the same for a little bit of a close-up for the yellow columns that has this information of chromosome position, mapping and score. We are not going to use this today, so no, not problem, but it's useful information. Then the green columns are a bit more important for us for today, because they have some stats like call rate that we're going to be using today, and also reproducibility. So to summarize, a relevant information you should remember is the clone ID is the unique identifier of the sequence tag, which corresponds to each sample set. Then you have a call rate, which is the proportion of samples for which the genotype call is either a 0 or a 1 rather than a hyphen. So it is important to know, especially call rate, because it's a good measure of quality. And then the reproducibility, that is the proportion of technical replicate assay pairs for which the marker score is consistent. So the reproducibility is a stat that diversity arrays calculates and gives to us. So you have some other information here that we're not going to use today, but if you want to know. So what is the program we use to analyze this data? we use the language R and the RStudio IDE. So basic knowledge in R and RStudio is needed to perform this analysis. I'm going to provide an introduction to it, but if you want to know a little bit, I'm going to also provide some information. So first, what is an IDE? An IDE is an integrated development environment that enables programmers to combine their activities to write software in a single application, editing source code, building executables, debugging and plotting, etc. So if you want to download R, you can go to this page and choose the link for your operating system, if you work in Linux, in Mac, or in Windows. In this case, I am working with Windows, so I downloaded this. Additionally, you have to download RTools, and it will depend on the version of R you are downloading, so you can download RTools. And to download RStudio Desktop, that is the program we are going to use today, you can go to this link and you can install it. So a note here, our RStudio used to be called just RStudio, but now you can also find it at POSIT. So if you see POSIT, they are from RStudio. So if you want to learn a little bit more, and if it's a basic, if you want to learn and you haven't used yet R and RStudio, I am giving you some links that you can use to learn for beginner friendly. We have the first link, then the R Ladies of Sydney, and also if you want to go and find your way in R RStudio Education. So to know a little bit more about this program, this RStudio IDE, you have some parts of it. You have 4 parts. The first part is the source here, and the source pane is for writing and editing your R scripts. Then you have the environment and history. In this page, this part will show your data sets and objects created. Then you have the console terminal. And in the console, you will see the execution of your code. And also you have a pane for files, plots, packages, and help. So these tabs allows you to navigate to your folders, also the plots you are producing, and if you want to know more about the packages. So some basic terminology I will be using today. We will be using the term R functions that can be also called commands. And the functions performs a task in R. So if sometimes I will have a function called copy, so because I need to copy something. If I want to save or load, so they have to perform a task. They take in inputs, called also arguments, and they return an output. So the function will ask for an input. Sometimes it would be a file or a name, etc. So another term to know is what is a wrapper function. And a wrapper function is just functions that call other functions that do the real work. So they're just a wrapper. So they are wrapper functions. Another term is R packages, that is the collections of functions and data sets developed by the R community contributors. They improve and expand the basic built-in R functions. Typically, a package will include code, and sometimes not only R code, sometimes Python or other language, and documentation for the package and the functions inside, some tests to check everything works and it should, and it's Okay, so I am representing a function as a page, and another package would be as a book, like a book. So it's just a collection of functions. Then we will have something called R libraries. The R libraries are just the directory in which R stores these packages. So it's like a bookshop. And the R package repositories are free online collections of R documents that organize these R packages. And we have CRAN and bioconductors as examples of these repositories. So we will be using these names: functions, packages, libraries, and package repositories. So what is DAR-R? We are working with DArTseq markers, but the specific package created for these DArTseq markers is called DAR-R. So it's a package for loading, filtering, and in general, manipulating and analyzing these DArTseq markers. It uses the format from the package ADGNet to analyze SilicoDart and SNPs. Please notice that many of the scripts are these wrapper functions for other already available packages. So DAR-R is based on another package called ADGNet. If you want to know a little bit more about the package, you can go to this link and learn a little bit more. I am also providing this tutorial guide. It's a little bit old, but you cannot find it online anymore because they created some other tutorials. So they have more tutorials, but I think that these 4 are the most important ones. If you want to know more about the marker, you can read the first one, how DAR-R stores your data and data inputs. If you want to know a little bit more about manipulating this data, you have the second and the third tutorial here. And the last one, if you want to know a little bit more about distance and visualizing population genetics. So to start this workshop, we're covering a little bit about R and RStudio and the package we're using called DAR-R. And to specify that DARTseq has low density markers and also high density markers. In this case, we are using low density markers. So we are not having 40,000 markers, for instance. We just have about 800 to 400 to 4,000 for low density markers. So for this demonstration, we are going to use a subset of 200 Zambian samples, so the demonstration is easier, but the original file contained 259 samples. We had to put them aside because of some genotyping problems. Please use updated versions of R on RStudio, because if you use all versions, some things might change and you will have some problems. The specific versions that I am using in this workshop are the R version 4.2 and RStudio version 2022-023. So what is the workflow of this workshop? First, we're going to start with an exploratory analysis. And after this exploratory analysis, we're going to analyze and improve our results. For the exploratory analysis, we're going to learn to load DAR-R, the library, and the SilicoDart data. We are going to do a quality analysis. After that, we're going to calculate the genetic distance between our samples. Then we're going to find clusters by using the neighbor joining method. And after that, we're going to plot the neighbor joining tree. When we finish that, we're going to improve the results, addressing some problems with the data. dropping individuals not useful for our analysis, but just putting them aside, not ignore them. After that, we're going to impute the missing data we have in our data sets, and then do it again. Calculate the genetic distance between the samples this second time. Find clusters by the neighbor joining method a second time, and plot the final neighbor joining tree. And finally, doing something called annotating the final neighbor joining tree. So what is the goal of this workshop? The goal of this workshop is produce this tree that has all the information we want. So this is the goal we're going to get here. So before we start our studio, we're going to provide the directory for the project. The name is this name. You can see Workshop 2023. And you need a folder called Files that contains the reports provided by Diversity Arrays. I am giving the name this, Silicon DART Sweetpotato, and the number of markers and individuals. And the plots, that is a folder that has the outputs that we will produce in this workshop. So a reminder that you will have this, and this is the most important information, that is the report of the Silicon DART report. And a little bit of a note here, because it's a problem that happens a lot, that if you are sending samples that may have the same name or the same code, it is better to give the sample a suffix, that it could be a letter or a number, because the program won't recognize the samples with the same name, right? So it's important that the names of your samples are unique, even if they are copies or different genotypes of the same sample. And another thing that is not provided from diversity arrays is something called an individual meta file. An individual meta file is a file that contains more information about the samples. In this case, it is compulsory that it has the ID with the names of your samples, and also a column called POP, that is what you consider the population. So if you are given more information, such as latitude and longitude, you should use the word lat and long. If you change it, the program won't recognize it. So for this particular data set, I am considering the population the same as the country. But you can, but this POP can be variety, a different phenotype, or any useful classifications such as fruit color or breeder. It depends on what you're considering your population for your analysis. Now, it is important that the names are ID and POP, because if you change it, again, the program won't recognize. Fortunately, for the dataset of Zambia, we have passport information. So this is very useful to know more about the samples. We have latitude, we have longitude, we have the collection site, we have the province, and even the elevation. So to start our studio, you should look for this icon. So I have it here. And this is our studio, right? So let me please order here this, so we can see better. So the first thing we need to do is create a project, right? You can create a new project, but you can create a project for a new directory, or you can create a project for an existing directory. Since we're going to use a folder that has the information we need, it's important that we create it from an existing directory. So we go And we can create a project with this icon here. And we have these alternatives, right? So we're going to choose existing directory. And we need to know where is our information, or you can just browse and go and look for it, right? So in this case, I have it here because I have here the folders for files and plots. So I'm going to open, I'm going to create the project. So you can give it the name that you want, but in this case, I am creating it as this name, SWP for Sweet Potato Workshop 2023. So while it's creating, I just want to clarify that for this workshop, you don't need to create any scripts or nothing. You can just choose the information we're given. And the only thing we need to do is copy and paste. So even the graphics we're going to provide. So this is to know. The first thing we need to do is create an R script. So how do we create a file script? You can go to File, New File, R script, File, right? New File, and R script. But a shortcut you can use, that I always use, is go click here in this blank page with a plus and go and do R script, right? So since we are going to use some packages, you will just need to copy this, copy it here in your script and run these lines. To run it, you can just put your pointer here, or you can choose all the line and click on run. Right. In this case, I already have these packages installed, so I am not going to run it again, but you should install it. You should run it all. Okay. An important thing to know about installation is that there are some different ways to install these packages, right? So you will see here in the pane for files, plots, and packages that you have an option here called packages, and you can see this icon called install. Oh, it's taking a little bit. I don't know why. Okay, and you can just type the package you need, in this case, .r. But I will recommend that you put the library you need, that you put the package you need in your script so as to not forget, because sometimes we don't remember the packages we use since we use a lot of them. So it is a good idea to have it in your script. And I will also recommend that you install .r from GitHub. And to install it from GitHub, you need to run these two lines. First, library that is calling the package that is already installed, the DevTools, that is called DevTools, because we need this function called install GitHub. So if you run these two lines, you will have .r from GitHub. It is important because some functions are updated here and are not updated if you install packaging from here. So since I already have it, I won't be installing it again. So we're going to start the exploratory analysis. The first thing we need to do is load the DART library. What is the difference? When we install it, we have that information, but we need to call that information. So we're going to be working with the DART library. So again, there are two ways to do that. Right, you can go ahead and in packages, you can search for .r and just click here, right? Right, to call the information from the package. But the other way that I will recommend, again, is having what you need in your script. That way you don't need to remember which library you use if you're working with different libraries. Okay. In this case, when we're working in a directory, in a project and a directory we know our position in the computer. But if we don't know, we can use this function called get working directory, and you can also change your directory, but it's not necessary for this workshop to change any directory. Just a note if you need it. And the first thing we need to do is analyze the quality of our markers, because we don't really know anything about our data set. So an important note here for .r, what we consider a marker, a silicon color marker, in the program is called locus, and what I consider samples are called individuals. So I will be using them as synonyms. So if you see locus, sometimes I will be talking about the markers or the silicon color markers. So we need to load the information that is in a CSV file that was provided by the company. And it is important that it's a CSV file. If you change the extension, the program won't recognize it, because these functions work with CSV files. A term you need to know is something called a genlite object. I know it's a lot of words, different words, but you will be seeing these words if you want to analyze DARTseq markers. And a genlite object is just an object of the format genlite. That is a class for storing in a compact way genotypes. In this case, it's normally used for binary SNPs, but it can be used for any level of ploidy. That's why it's very useful for the Silico Dart, right? So the genlite objects are normally used in this package ADGenet, but DART-R is based on this package ADGenet, so they work with genlite objects. So we need to load the information we have, and the first function we're going to use is called GL read Silico Dart. DART-R has very descriptive functions, so this GL just means that it's a genlite format, right? So this function GL read Silico Dart needs an input. So the input for this function, the first thing is a file name. That is the name of the CSV file containing the Silico Dart data. You can use the name that diversity arrays give you, but you can also change it. I prefer names that are more descriptive. In this case, I am giving it a name, the marker Silico Dart, Sweetpotato, that is the species, the number of markers, that is 736, and the number of individuals of my file, that is 200. So I prefer those names as to know more without opening the file. The other thing is the top skip, the number of rows to skip before the header row containing the specimen identities. If you remember, we had a number of asterisks. So this top skip just asks you for the number of asterisks to skip, right? You can see here, the top skip is 6. And then NAF, that is just a missing data character. Again, it could be a 9, it could be a 99, but for DArTseq is a hyphen. And the int meta file that was not provided for the diversity arrays, but we have, that is the name of this file containing the metadata assigned to each identity of individual. And as you remember, the compulsory was the name of the sample and the population of the sample, that in this case is the country. So I'm sorry, just 1 moment. So we need to just copy this and put it in our script. Right. So we can just run here. And the program is started. Right? Here in the console, you can see the program running, and it gives you some information, right? How many sequencing tags has, how many samples it has. It say that you're giving it 200 identifications, IDs, or names of individuals, and also the information you're adding for your for this file into meta file that has the population, and for the Zambian sample you have province, you have elevation, etc. So you can see the results here also. I am adding here what the console is saying. And a little bit of a note, diversity arrays normally give clone IDs, unique clone IDs, but sometimes it can have the same name, so the function corrects it and gives us a suffix of 1 or 2. It won't normally happen, but again, the function will correct it. The second step is to save our data. Why? Because this time, this read of our files took like a minute. But if you have a very, very big data set, like 40,000 markers, I don't know, it will take a while because it needs to fashion this information with the information we have, that is being compressed in a single object here, and it is converting it to another format called JSON, so it can take a while. It is important to save it. You just need to run this to save, and it's already saved. So something I didn't explain that I think it's important to know. It is, I mentioned it, but to remember, this is a function, and the function needs an input, and we're giving it an input. When we use this character, that is like an arrow, we are saying it, please store this information here. When we store information, this is what we call an object. In this case, this object is a JSON object. So when I talk about an object, is what it stores all I gave it before. So if we need to use this information without these steps, we can use the function load. So if you are finishing for the day and then you need to do it, you need to do the analysis the next day, you will need to do this part. You just use the function load. Yes. So to know a little bit more, we can run this to know the names of the metrics that diversity arrays give us, right? Because you can see this in an Excel. You can go ahead and look for it in an Excel. But again, sometimes you will have so much information that your Excel will open in half an hour. So sometimes you just want to know some things. In this case, if you run this line, you will see the information that your dataset has, like the clone ID, the allele sequence, the trim sequence, the one ratio, call rate, etc. And if you want to know the individual things, the names of your samples, you can run this line. And you will have here, this just means that I am asking just for the first 10 samples, the name of the first 10. You can change this number to know more, right? 100 or 200, because we have 200 samples. In this case, if I want the 15, I will have more here in my console, right? For everything, you have the result here after the line I run. And that's it for loading information. So now we have our information in Dart, in RStudio. But we don't know anything yet about the information we have. Is it good data? Is it bad data? We don't know. The first thing we need to do is produce a report of the data quality. How do we produce the report? We start with the monomorphs. The monomorphs are markers that are identical for all our samples, therefore are not informative. This means that for SilicoDart, and if a marker is a monomorph, it will be a 1 for all the samples, or it will be a 0 for all the samples. So it's really not useful. So if we want to know, we just copy this and we run it. So again, I am using this, a function called GL report monomorphs. The functions are very descriptive. And I asked them to know this monomorph about this object. This object is the object we have, the main object of the information that contains all the information we need, right? So we run this. And this is the result. We have 736 markers. Out of them, 712 are polymorphic, but we have 24 that are monomorphic, 24 that are not useful markers for us. Right, so we know a little bit more about our samples. Even for low density, 736 is a bit low. And we have 24 monomorphic losses, so it's good to know. So if we need, we will erase it. After that, one of the most important filters, it would be the call rate. So we need the report for this call rate that, again, is the proportion of samples that for which the genotype call is either a 0 or a 1, rather than missing data. So to know the call rate, we just copy this, right? So we need the call rate report. And I'm asking this function called report call rate for the information we're giving it, that is GL Silico Sweetpotato. And in this case, you have another option that is called method. And you will see now why it is necessary. In this case, it's log, because for this package, log is marker. So I am just asking it for the call rate of the marker. So this is the result. This is the result. And you can see here the distribution of the call rate. Most of our samples are good, are above 0.85, which is very good. But we have some of them that are below, but in general, this is very good for the marker. We have even some that are 100%, so they don't have missing data, which is good. So you will also have more information here in the console that again gives you number of markers and individuals, the minimum, that is 0.83, that is not bad, and the quantiles, right? And the mean, that is 0.94. So this data is good. It's good. So this is just a cumulative, and it's a table just to teach you to see how many you will retain if you filter, right? So if you put a threshold in 1, you will just retain 143 markers, right? So you can, maybe not a good idea to be that strict to just put the threshold in 1, right? But you will have, maybe if you want to put the threshold in 90, you will retain 592. That is the 80% of your sum of your markers, right? So this is just indication of that, this table. You have it here also in the file of the workshop. You have this information, and you also have the plot. So a call rate for the 730 markers is above 0.83, so we have good quality of markers. The other thing about call rate is the call rate per individual. Why it is important? Because sometimes it's not the markers, but the individuals that are not performing very well. Maybe we sent bad DNA, or it was degraded. So it is important to also know the call rate of the individuals. So we just run this line. The only thing is changing is the word is for method. For markers is log, and for individuals is int. And again, you have here your results in the console. It's giving you the minimum, this 0.86, which is good, and the mean of 0.94, which is also good. And then it will also provide the call rate per individual. So we have a lot of them with 99, 98, 97, 96. So again, all of them very good. And it will give you the last one, that you will sometimes find that you have some of them that are in 0.50. So you will know which of them is. So if you want to make a list to erase them later, so you will have it. In this case, we don't need to do that because we have good call rate for every sample. And you can see here also the plot in plots. And you can zoom, you can choose zoom, and you will see the distribution. Again, we have good data, just some of them are below 0.90, and a lot of them are very close to 1. Another report, another more information we need to know is the reproducibility. So again, the reproducibility was provided for us for the diversity array, so we don't need to calculate anything here. It's already in a CSV file, but we need to know a little bit more about it. So we just produce the report, and we will just run the line. And this is our result. And very good reproducibility, a lot of them in 100, and a little, some of them in 98, and some of them from 95 to 96. So again, good reproducibility. And this is the result in the console. The minimum is 0.95, and the mean is 0.98. Okay, great. This is the results if you want to see it a little bit more. So the reproducibility is important, but because it gives accuracy to the analysis and allows you to take the same results every time you run it. So it is important to know reproducibility and not only call rate. So now we know We produce the reports and we know a little bit more about the quality of our data. The conclusion is that it's good. It's very good. We don't have many markers that are of low quality and neither individuals of low quality. So now we need to start the analysis. Now we know all this information. So a thing I do, but it's not necessary if you don't want to, is I clone the information because this is our original data set that has the information we load in the first step. But sometimes there are some steps that transform the object in another object. So I don't want to lose this information because it's my raw data. So it's not a good idea to touch it. So I prefer to work with the copy. So the only thing I am doing here is storing this with the arrow and in this object that I am calling GL2. It's also a name easier to work with. Right, so I am storing it in GL2 and I am running this line. That's it. Now that we're working here, you can see this environment pane. Environment in history. History tells you all you've been running. Every line you run is here in history. And every object you created is here in environment. So you see our data set that is a large young light and the weight of it, and the clone I created that is called GL2. Right? So you can see every object here in the environment panel. Now, we have good data, but it is necessary to know how to filter markers. If you remember, when we produced the report, we have 24 monomorphs. Maybe you can say that it's not that much, but again, you will sometimes need to remove a lot. So we are going to take away the non-informative markers. And we just need to run this line, that is GL. It's not reported anymore, now it's filter and monomorphs. And I am asking the function to filter from this object, and it's 0 because the difference it has to be 0 for all of them. It's equal for all of them, so that's why B equals 0. And again, I run this line, and now I have an object called GL3. Right? But how do I know if it worked? I run the next line that is called number of locus, right? N log of the object. And here you can see the result is 712. Right, so yes, it erased the 24 monomorphic markers, so we're good. Now the other thing to do is filter the reproducibility. We just need to run the line again. So since we have a particularly good data set, I am putting the threshold a little bit higher, that is 0.96. And I am storing in the object called GL4. GL filter reproducibility of the threshold 0.96. I run it. And this gives me a plot. So this plot is just the representation of the filtering, that it used to go from 0.95 to 1, and now it goes for 0.96 to 1. Right? That is in the plot. But again, you can ask how many markers we have. And now we have 665. So the filters are working. And we have now we need to filter the call rate. So we just run this line. Right, that is a little bit, it has more input. So we are going to analyze this input that the function needs. We're using the function GL filter call rate from this object that has passed through filtering of monomorphs and through filter of reproducibility. And I need to filter the markers. That's called log. And I'm putting the threshold of 0.90. And we have one more that is called recalc true. Why I'm using this? Because when you filter markers, the stats will change. So you need to recalculate them for the data, for the filtered data. That is what I am using recalc true. I run it, and it's completed. Very fast. These also produce a plot. The same as reproducibility, it used to go from this 0.83, I believe, to 1, and now goes from 0.9 to 1. We can use the same function we were using in log to know how many markers we have left. We have 575 markers. So although we are reducing the number of markers, but 22 doing this filtering, the quality of data is improving. So it's a good thing to do. Now, one thing you can add if you are working with a big data set is add this line called plot false that you would put just here. So in that way, you don't have to, you don't produce this plot here that we've seen. Why? Because sometimes it takes a while, and sometimes we just don't want to see it, right? Sometimes information from the console is all right. So we're finishing with the filtering, and an important thing to do is saving this information. Why? Because again, we don't want to pass through the process again every time we want to do an analysis. And so it's a very good idea to save it, right? In the function save, where I am giving it the object I want to save, that in this case is the one that has passed through filters. And you have the name of the file here. In this case, I am saying to the function that I needed to save the information in the folder files. And this is the name I want to give it. You will normally save objects with these extensions called RData. Again, I am very descriptive with the names of the files. So I am giving it the name of the object, the number of markers it has, and the number of individuals of this particular object. So I just save, that's it. When I need it later, I can just load it with the same, with this line. Okay, so geographic origin. Sometimes you won't have this information. You won't have the information of latitude and longitude, but fortunately, in this case, we have it. So we just need to run this. Maybe we can put here in a comment. Right, we need the library maps. And we have this function called borders. I am just asking the function to search in the world, the region, sorry, we don't need this, the region Zambia, because this data set is just from Zambia. Maybe we have another country, we will need maybe Nigeria or later Madagascar when we have the information. But for this, it's okay to have this map. Now, if you remember in our int meta file that has the passport data, we were provided with latitude and longitude. So we need to read again this file. It's the same file we gave the first function we use. So nothing needs to be changed, and we just run. I am storaging this in point Zambia. And then we have the next one, that is a copy and paste. You don't need to change much. So I am going to look for the library ggplot that I need for this. I am asking the function ggplot that use this information of the map, called map Zambia. And then I am asking them to please put dots, points in my map. From this data that I'm given, that is stored here, from latitude, for longitude and latitude. I want the color of the province because this is just one country. If we have maybe 4 countries, we can use just country. But in this case, since it's just one country, I am using another administrative subdivision, that is the province. So I will just run this, right? And then, sorry, I'm gonna run this first. And this is my map. So I can see the distribution of my samples here. And I have some samples for the Copper Belt province, also for the Northwestern province, for the Eastern province, and the Southern province. So our samples do not represent every single corner of the country, but this is the distribution of them. Right, so I am just giving you some information about appearance, like the x-axis label and the y-axis label, the legend of the title, and the theme of, this is mostly for background, if I want a light background or a dark background, right? And this is just to maintain the proportion of the map. So I am adding here the map of each province. So you can see that for the Northwestern province, we have just a little bit of the Northwestern province here. So it's very close to the Copper Belt province. So those might be quite similar, but we don't know. So, okay, we are finished with the geographic origin. We know a little bit more about samples. And now that we filter all the information we don't want, we need to find the genetic distance and plot our neighbor joining tree. We are doing it today with the DART function because DART has a lot of very useful functions, and we can do everything with just this package, DART. But you can do it also with other ones like vegan and popper R. But we need just DART. So we have 2 options here. We can use the option GL_dist_pop, again, very descriptive, from a distance from a GenLite object between populations. So now we have in population only samples from Zambia, so we don't need to do this. But most important, the distance between individuals. So between our 200 Zambian samples, we will need to use the function GL_dist_int. First, to calculate the genetic distance, we are going to run this line. right? So maybe we can specify here in a comment that is the genetic distance. So what is this function doing? It's just I am giving it the information, the filtered information, and I am asking for the method Jacquard. So the distance is going to be Jacquard distance, and I am giving it the name Jacquard Silicodar for 200 individuals. I am going to run it. Okay, it's finished. We have here the result. We have the result here. And this is the distributions of the distances, inter-individual Jacquard distance. We have some of them are very different, right? We have just few of them that are close to zero. You have here also the result. Okay, so we have the distance. After we create the genetic distance, as we saw in the workflow, we need to find the grouping of our samples. So we do something called clustering, because we need to find the clusters. And the cluster method we're going to use is the neighbor joining. So the principle of this method is to find pairs of operational taxonomic units, that in this case is individuals, that minimize the total branch length at each stage of clustering. So it starts with the start and then it constructs from there. Again, if you want to know a little bit more about the Jacquard distance or the clustering method they were joining, you can go ahead and read a little bit more from the tutorials I am sending. It's very good information, very specific information as to why we use this distance and these clustering methods. So to do this clustering, we are going to use the library ape. Right? So we just run this line and we have now the library. And from this distance, the Jacquard Silicodar 200 individuals, I am asking for the neighbor joining clustering method. I am running. Okay, now we have this object that is the clustering of our samples. But then now I need to observe it. I need to produce a plot. I am going to, this we're going to use with a library called GG3. So I just call GG3 from library. That's it. Now I have that library. I'm going to run the next line. That is plot a basic tree. Now, the function from GG3, this object that the input 3 silico Sweetpotatos, SWP, is the one that has passed through calculating distance and clustering. If you give it any other information, it won't work. And then this plus GeoGeoLab, this line only works for the tip lab. I am asking GG3 to please give me the names of each sample at the end of the branch that is called the tip. The color I want is black, and the size can change according to what you want. In this case, I'm putting 1.5 and just run it. And I have already my result. You can see that it's identical. Here it is. But we don't see it very well, right? We cannot analyze this. I don't know which sample is this. I cannot see. So one option is to zoom to see, but again, it's not that clear to me which sample is which sample. So what we need to do is save this plot. So I just need this line I need to run this line. So I am saving this dendrogram in the folder plots. I am saying that it's from Zambia, from 175 markers, from 200 individuals. And the width and the height can change according to what you want. But in this case, 8 and 20 works very well. So I will just save here. So it is saving 700. So it is saved now. Now, a little something you should know about this function gsave is that it saves the last plot you produce. So if you produce another plot, that is the one that it will save. It doesn't work with the name of the plot. It just works the last one. So if you, sorry, I want to show you the plot, but it's taking a little bit of time. Here it is. So this is what we save. And now we can see some things, right? Now we can see our dendrogram, our samples here, right? That's it. Very good, very good. It's okay, very good. Very good, very good. But now we have a problem, right? We see something very, very odd here. We have something very odd here. That some of the branches are the other way around, right? That is first problem, right? The other way around, the other way around. And we have some samples here that are kind of functioning as a root. So it's a bit odd, right? So we will need more information about these samples to really analyze them, right? So one thing we are doing now is sending them to know if maybe the DNA quality was not good enough to produce good results, or maybe the samples are just like that, but we need to verify them. So for that purpose, we will need to maybe cut them from this particular analysis, right? So I have here, if you can see a warning message. Now, a warning message is not an error, but it's something you should take into account. That your tree contain negative edge lengths. As you saw, you have the branches going the other way, right? So this is a bit of a problem. It might be caused by too much missing data or other things, but we can, maybe it's being caused by those samples that are dragging the information. So we need to put them aside for a bit so we can analyze them later. So this is the suspicious individuals we are going to drop for today, and we need to analyze them later, right? So erasing these problematic samples for today. To do that, you need to create a CSV file with the names of each of the samples you don't want for your analysis for today, right? So in this case, I am adding all this information in a file called samples to drop. Samples to drop. Okay. And you just need to copy and paste. So this function called read lines, I am just asking for this to read every line of this list I created for samples to drop from CSV file. And I am storing in an object called samples to drop. So samples to drop is my list. The function is gl drop individuals, and very, very descriptive. from our object, and the list is this list that I read, that is samples to drop. And recalculate, yes, because when you lose markers, when you filter markers, you need to recalculate the stats. But when you lose individuals, you have to also recalculate the stats. Oh, I'm sorry. I think I didn't copy the file. Just give me 1 second, please. Just 1 second. I am going to copy the file to the position it needs to be. So it's here. And in files. Oh, very weird. It's here. I'm sorry, I don't know what it's not working. Ah, okay. It is like that. So samples to drop is already read, and I can drop individuals. Okay, that's it. So now I am dropping the individuals I find suspicious, right? But the other thing we can do is input what the missing data we have. Now, it can be controversial to input data, but especially when you have a lot of missing data. But every algorithm inputs in some way. So in this case, we are going to use the function GL input. And this will just input and put it in an object called GL, but inputed. Okay, it's completed. Now we have input data. So what is input, right? It fills the missing data with what is probably the correct. It is a 1 or is a 0, right? You can do this imputation when you don't have that much missing data, and since we don't have that much data, we can do this. And now that we've improved our data set, we need to repeat what we did the last time, that is calculate the distance. So we calculate the distance, but for our samples that are, for our object that has imputed information, the method is still Jacker, so you just run. And now it's calculating the distance for our improved data. And you can see the samples that are closest to 0 are less. This is the distribution now. And then we need to cluster to find the clusters of our information. So we just again copy and paste the neighbor joining for the imputed data. There it is, it's already done. And now we will need to plot the information. So we plot the information, right? Plot, how do we plot? Again, it's the same line, only that I want it from the imputed information, right? So I just run this. Okay. And that's it. And this is our data, right? But maybe that we improve the information. We don't want to see it only in black, right? So something we're going to do is called annotating the tree. What is annotation? Annotation for phylogenetics is basically give color, format, and style, or in general, coloring the branches or highlighting clades, right? Since, I don't know if you remember, but the goal was to produce a dendrogram that has these colors. So we're going to need these 4 libraries. We're going to need GG3, A, and dplyr. And again, if you remember, we have a meta file that has all the information from our samples, and we need to storage in info. But why would you say? Because we filter things. So we have less individuals. Why are we using the same meta file? Because fortunately, it will take only what it needs. So if we have information for 1,000 and you're only plotting for 200, it will take just the information that it needs. So it's okay if you use the complete information. Now, to give it format, to annotate the tree, we're going to copy this. Now, it's a little bit more complicated than the first one that has only 2 lines. But it's very, you can understand it just by reading it. I need the GG3 to plot the tree that is imputed. And the layout I am asking is rectangular, but this layout can be circular or can be a fan, for instance. But in this case, I want the rectangular. I want this to add the information that is here in info, because I want to color by the province. So that is why it needs we need to add info here. Again, I want province at the point. I want province of the branches, GM3. So the color of the branches, here GM3 is for the color of the branches. I want them to color by the province. And GLab, that is the names of the samples. I haven't changed this. And in this case, I am giving it manual color because I want to give any specific color to each province. In this case, I have Eastern province and I want this color. And for Southern province, I want this color. And for Northwestern province, I want this color. And that's it. So, run it. Yes, there is still a warning message. But you will see here in the plot. Here it is. Now we can see a little bit more, right? So this is my legend. The Eastern province is this blue, green-blue, right? So all my samples are here from the Eastern province. Then we have some of them for the Southern province. Southern province, right? Then we have the Copperbelt province. Here we have some samples that are only for the Copperbelt province. And some of them are here, the North and Eastern province, right? It is not a clear separation, maybe because it's inside the same country. And you can say that they share a lot to birds between them, right? There is some of them that are most for the Southern province, right? So you can analyze a little bit better by province with this annotated tree. Now, if you see there is still a warning, right? Because if you see here, this information used to be the ones that had the branches to the other direction, right? So after passing through imputation, you can see here that maybe those are the same sample, right? Because here the distance is almost 0, right? So maybe this distance that was the other way, since it's imputed, is just here, right? So this is what the warning is saying for us. Okay. So now we have an annotated tree. Another thing you can do, if you really don't know anything about, you don't know anything about colors, you don't really know the provinces, or you, so if you don't put that information, maybe to have a little bit of less lines to remember, you can run it, without the color, and it will give you automatic colors, right? And it will give you the legend. So you can also have the automatic colors. And since we produce this, we can just save it. Right? Oh, sorry. So we have 155 colors. 155 individuals. So I am saving it with this name. I'm going to save. That's it. Okay. But our goal was not a rectangular dendrogram, was a circular dendrogram. So how do we do that? Right. It is the same lines, it's exactly the same. The only thing you will need to change is the layout that I wanted now to be circular. So I run it again. I run it again. And this is the plot. This is our final goal. That is producing these dendrograms with information of the province. Right? Maybe we can save now to see it better. So here it is. We just need to run the line for saving. I am specifying that it's circular, so as not to confuse it with the other one because it has the same information. I have it already saved here, and you can see a little bit more, right, in this circular format. Right, but we can just run it again and save it. And you can see that after imputation, we have these samples that were a little bit weird, a little bit more ordered now. So it's in fact improving what we had at the beginning. Now this function gg3 has it can do a lot of things. If you want to know a little bit more, you can click on this link and you can learn more about gg3 and doing these dendrograms. A note, just to finish, is that you can also use the GL this population. I'm sorry, here in Sati. But we in the beta file have a population to Zambia. But you can redefine the population to whatever other information you want. In this case, I want it from province. I am just cloning the imputed data so as to not modify it. And I am putting in GL7. And if you run this, now your new population will be province. And you can run this line to know the distances between the provinces, the 4 provinces. So this is the end of the workshop. You have here the table of information. If you want to repeat something, you can just go ahead and look for it and click. I have added a part called Beyond this workshop That is, if you want to work with the SNPs, you have the 2-row SNP-based data sets and also the 1-row SNPs data sets. So a little bit more information about that and how to do this analysis again, but with the SNPs, right? How you can do this and how you can see the information from the other marker that diversity arrays produce, right? Some other filtering that SNPs may need, right? A dendrogram with the SNPs. And another thing that is the principal coordinate analysis that this workshop won't cover, but you can learn it from this file too, right? You can do it for silico dot markers, and you can also do it for the SNPs markers. That's it. I am adding some other options if you don't want to start with R on RStudio. You can use something called the Dart view, right? And you can use also use key KB compute. Okay, that's it. That's it.
Thank you, Veronica. This was awesome. What I want to emphasize to the attendees, if you're totally bewildered and lost, you're not alone. It looks complicated, but the workshop was set up as a cut and paste. We recorded the workshop and that recording, as well as the presentation, will be available to you within an hour or two, and you can go through it and try this. What I suggest, and Veronica, could you remember we ran the Zambian data in a dendrogram with some of the other African countries? Is it possible for, while I'm giving a little introduction, for you to call that up? I couldn't find it on my computer. And so you can show them the potential to do it that way too. What I suggest is that anyone who wants to try and do this with their data set, to plan on at least a 4-hour session your first time, because that's how long it takes. It's not that complicated once you get started, but like anything, riding a bicycle, you've got to practice before it gets easy. And remember, this is an introduction to some of the tools that are available. These are the very basic tools for analyzing genetic diversity. And what we've provided, what Veronica's provided, is a step-by-step way to do it. So when you try to do it, please feel free to get back to us. Everyone's got my email, or Alex or Vanya or any of the other authors, and we'd be happy to try and step you through it, or at least put you in contact with someone that can help. I also wanted to mention that if any of the attendees want or need a certificate of attendance for the workshop, please let me know, because we'll be happy to prepare one and send it to you. I think right now we can open it up. Faye, I'm not sure how we do this, but open it up to any questions, whether we do that through chat or otherwise. But does anyone have any questions at this point that they'd like to ask, or Veronica, or anyone else? And I'm just looking at chat to see whether anyone's there.
Can you see my screen?
I can see your screen. Permission needed from meeting host. Okay, yes. So if anyone has any questions, please let me know. Let us know. Okay, what Veronica has put up, and do we have a legend for that we can see, Veronica?
Sí.
There you go. Okay, then we can go back out. What Veronica has put up is, remember when we calculated the genetic distance by population, we did it by province. Here, it's done by country. And what we have is Sierra Leone, Ghana, Zambia, and I'm not sure whether we have Mozambique on there as well. But here we can see that the sweet potato land races within the African continent appear to not 100%, but pretty much segregate by country, which is very interesting. Remember, we saw when we looked at the provinces within Zambia that there was some segregation, but more or less they were mixed. And that's probably because the land races are exchanged within the country. But what's interesting is it doesn't look like, now these are great distances, it doesn't look like they are shared so much between countries, at least for the countries we've analyzed. Now, what we'll do for the Darwin project is once we get the information, the genetic DArTseq information from Madagascar, we'll be able to compare Madagascar's genetic diversity with Zambian. I would predict that we'll see great segregation. The two will segregate by country. But then what we can do is we can compare this, and this is where the analysis gets more and more complicated, with the SIP collection, and we can actually start to maybe try and make predictions based on genetic distances of similarities between the different continents and the different countries within the two continents, that is South America and Africa. But also there's samples from Oceania, so we can see where shared genetic information is between these different countries. So with that, does anyone have any questions, or do you want me to continue with the questions?
We have a question from Rainer. His hand is up.
Perfect. Sorry, Rainer.
Hello, Dave and Veronica. Great presentation. Thank you very much. I've learned a lot, I hope. I have two questions. Can you expand a little bit or give us some guideline about the concept of call rate and reproducibility? You talked about the thing call rate. One is for marker, the other is for individuals. Is there some let's say guidelines, some recommendation value where you should say, okay, this is acceptable and this is we should remove it, or is it difficult to say it at this point?
Yes. Well, yes, call rate is something that I think it's always the first question, because it tells you how much information you actually have. So if I have 100 markers and 50 of them are presences, 40 of them are absences, I have 10 that the technique was not able to determine if it was a 0 or a 1. So my call rate would be of 90%. So I have 90 of these 100 that is information for the marker. So it will normally depend on your data set. Sometimes you will have very, very good data. So you need to try things. So you can start with maybe filtering 20% and see how your data behaves. Sometimes you can exaggerate and you put 100. I only want samples that don't have 0 missing data. And you will see that may be not such a good idea because you will lose a lot of markers. So you need to prove you need to try different things so you can find a balance. Now, in reproducibility is something that is provided for us. So the test is done in diversity arrays. And they will normally give you all your markers above 0.95%. That is what they consider markers of quality. So you can also, again, try a little bit if maybe your samples improve or not. With this, you need to again, a try, right? You can start with 5%, 10%, 15%, 20%. And sometimes you can start just with not filtering anything to know what you have, and maybe it's already good. So the filtering is just to go and be improving what you have.
Yeah, I think, Reiner, basically the higher the better. But as Veronica showed at one point, you have tables that will show you how many markers, for example, you lose when you go to a lower call or a higher call rate. So it's important. It's also important to know that anytime you filter out data, you're going to lose markers. And when you're doing these types of analyses, there's a point where you don't have enough markers to adequately say anything. So you always want to make sure that you don't completely eliminate all your markers. And Veronica gave an example there that if we asked for too high a call rate, we would have only had a few markers to analyze. And so that's another variable you have to look at, is how many markers you have left. Make sense? Did that answer your question?
Yes, completely. Thank you very much.
Okay. Okay. Do we have anyone else that has any questions?
I don't see any other, Dave.
Okay, I think an important point for everyone to remember that Veronica mentioned very early on, that for our samples, we can always put an A, B, C in front of them. And that's critical because often often it is recommended that when you send data to diversity arrays, that you have replicates of the same DNA or different DNA from the same samples when you do your run, so that then you can tell a threshold, a number of a statistical threshold of when things are the same and when they're different. Now, Veronica showed us those two samples that are very genetically similar. The question is, are they the duplicates of the same landrace, or are they different landraces that are just genetically similar? By putting replicates, when you're running it, you can have a statistical method of saying whether they're duplicates or not, because you can get an error rate between the running of two of the same samples. So that's why it's important to put an A, B, or C in front of each sample to make sure it's unique. I think that's an important point that people may not have gotten. And Veronica, in the dendrogram that we generated in the training, you said that those samples at the bottom were suspect. Is it possible for you to elaborate a little bit more on why they were suspect? Because they look like they segregated out fine, but I just wonder what made you think they were suspect. Yep, that's it. You're muted.
Oh, okay.
There you go.
So you can see here the behavior of your samples, right? That this is the like a normal distance between them, right? So even some of them are further away, right? So even the ones that are very close have say certain behavior, right? So that is for most of the samples. But then when we get here, you can see that even this is a cluster. But for this, I don't really see that they cluster. They are just accumulating in a line like this. So I think we need more information from the samples, right? I would expect that if these were a same cluster, they would be here like the rest
Okay.
right? Like the rest of them.
So it's because they're more flat that you are suspect of them.
Yes, they are flat.
So for more information, would you go back and look at the data that you have, or would you assume that you need to run new DNA for those samples?
I would prefer to run again the DNA, because sometimes bad DNA can cause a lot of problems. Some of these problems is that they don't match their pair if we are sending replicates. And in low density data, we're working today with low density data. We have sometimes a problem where the technique recognizes something that should be scored as missing as a 0. So these kinds of problems, it is, I think, better to repeat the DNA, but also you can do the analysis again and to see if maybe those samples are like that and they are different. So it's not that we are just erasing them and we don't want to know anything about them, but that we need to analyze again, to analyze further.
And when you drop them, you just drop them from the analysis. That all that data still remains there, so you can always reanalyze. And the way you renamed every time always allows you to go back to the original file and rerun it with different parameters.
Yes.
Yeah, I thought that was pretty useful tool. Okay, does anyone else have any questions, comments? So everyone is eager to go out and try the learning that they got today from the workshop. Okay, well, thank you, everybody. Faye, thank you very much for your support in getting this running. Again, we will get back in touch with you today with the recording and the presentation so that you can start to run and try this yourself. And as well, if anyone would like a certificate of attendance, please let me know. With that, I think I will say goodbye. And thank you all, and everybody have a wonderful day or evening, depending on where you are.
Thank you. Thank you. Goodbye.
Goodbye.